Welcome to Midday Scenario. In this video, we discuss the steps of acute inflammation. Now, what is inflammation? Inflammation is a response of vascularized tissues to infections and damaged tissues that bring cells and molecules of host defense from the circulation to the sites where they are needed in order to eliminate the offending agent. Now, steps of acute inflammation can be divided into two categories. One, the vascular events, and secondly, the cellular events. Now, vascular events occur before cellular events. Under vascular events, we will discuss vasodilation and increased permeability of the blood vessels. Now, vascular smooth muscles cause vasodilation, and increased permeability occurs due to endothelial cells. Under cellular events, we will discuss the emigration of leukocytes, macrophages, and phagocytosis. So, we all know that during inflammation, histamine is released at the inflamed site. Histamine acts on vascular smooth muscle cells. Vascular smooth muscle cells contain H1 receptors which are activated by histamine, and this causes increase in cellular levels of calcium, cyclic GMP. This occurs through the IP3 and DAG second messenger system. So this actually causes increased secretion of nitric oxide, which is actually responsible for the vasodilation action. Now, due to vasodilation, there is increased blood flow in the particular blood vessel at the inflamed site, and this will cause erythema or the redness at the inflamed site. Now, histamine does another thing. Histamine will also act on the endothelial cells of the blood vessel, and they cause release of intracellular stores of calcium, which will cause contraction of the vascular endothelial cells. Now, if the vascular endothelial cells contract, they actually increase the permeability of the blood vessel by increasing the interendothelial spaces. This causes fluid leakage. The fluid means the plasma fluid leakage. So this fluid leakage occurs through the interendothelial cells spaces, and this causes stasis of RBCs, because now, since the fluid is lost, the cellular count increases, or the hematocrit increases. That is, the percentage of RBCs increases at the particular site. Now, if the percentage of RBCs they increase at the particular site, so they actually push the WBCs or the leukocytes. This is because RBCs are far more in number than the WBCs. So this is called margination, in which the WBCs are pushed to the periphery due to the clumped RBCs. Now, after margination, there's an interaction between the WBCs, especially the neutrophils and the endothelial cells. The receptors on the endothelial cells, called E and P selectins, will act with the ligands on the neutrophils. So there is a binding between P and E selectins and the oligosaccharide ligands on the neutrophils. But this is a not very strong bond, and it kind of detaches and binds, which leads to rolling of the leukocytes, especially the neutrophils, on the endothelial cells. Now the next step is adhesion, in which the neutrophils bind firmly with the endothelial cells with the help of VCAM1 ligand, which is on the endothelial cells, and VLA4 receptors on the neutrophils. So in this process, it is firmly bound to the endothelial cells, and this is called adhesion. Now next, what happens is that after adhesion occurs, there is another interaction between the neutrophils and the endothelial cells, and this interaction occurs with the help of CD3 or PCAM1 molecules on both the endothelial cells and the neutrophils. PCAM stands for platelet endothelial cell adhesion molecule. So this process is called diapedesis, in which slowly the neutrophils will leak out through the interendothelial spaces with the help of interactions between the PCAM1 molecules on neutrophils and endothelial cells. Now, under the action of certain chemokines, there's a process called chemotaxis, in which the neutrophils is kind of attracted towards the inflamed area, or the area where the tissue destruction has occurred, or there's a microorganism. Let's suppose there is a microorganism which is present, and neutrophil is attracted towards that area with the help of chemokines. Now, neutrophils contain certain mannose receptors which will bind to the mannose residues on the microbe. So this binding occurs. And next, what happens is that when this binding occurs, there is a phagocytosis reaction initiation of the microbe. Now let's discuss phagocytosis process in details. After the neutrophil binded with the microbe with the help of mannose receptors on the neutrophils and the mannose residues on the bacterial cell wall, there is pseudopodia formation, in which the neutrophil kind of, or the macrophage kind of, engulfs the microbial organism. This is called pseudopodia formation. After which the plasma membrane kind of pinches off, uh, and this process is called phagosome formation. It is kind of similar to the endocytosis process. So there is a phagosome formation. The phagosome is actually a kind of bubble which is kind of like surrounded by the same material as the cell membrane is made of, and inside it is contained the microorganism. Now next, what happens, there is a fusion between the phagosome and the lysosome. Now we all know that lysosome is the cell organelle that is responsible for digestion of contents. So this phagosome fuses with the lysosome to form phagosome, and this phagosome will cause digestion of the microorganism with the help of reactive oxygen species and nitric oxide. So that was the process of phagocytosis. If you like this video, please like this video and share it and subscribe it. So that's all for today. Thank you for watching.